Welcome to the overview of the curation process in DisProt. This is the first part of the training for curation in DisProt. I'm Federica, expert curator and training manager of the resource. These are the learning objectives of this first part of the training. Describe what IDPs are, how to log in DisProt and what is DisProt, how the curation work in DisProt is credited using APICURON, list which are the essential information required for curation in DisProt, as well as describe the importance of rigorous data curation in DisProt. And finally, how to perform the curation process in DisProt, which are the steps to perform it. So let's see now what are intrinsically disordered proteins. Intrinsically disordered proteins, also known as IDPs, and intrinsically disordered regions, also known as IDRs, are proteins that do not form stable three-dimensional structures. They lack in hydrophobic amino acids, therefore they cannot form an organized core. What about the functional aspects of intrinsically disordered proteins and regions? In the past, it was believed that these proteins didn't have active functions, but now we know that they are actively involved in cellular functions. Moreover, inside intrinsically disordered regions and proteins, we can find short linear motifs that facilitate interactions with other protein partners. So IDPs and IDRs are involved in signaling, regulation, and many other cellular processes. Finally, they can also undergo post-translational modifications that enhance their functionality. What is DisProt? DisProt is the database of intrinsically disordered proteins. What you're seeing now is the homepage of DisProt. You can see the link to the resource here, and some relevant features that you can find in the home page. Here you can see entry examples of some annotated entries of DisProt. Here you will find the datasets available in DisProt. So the thematic collections from different biological areas that we collect in DisProt. Here you can find the login button to start curation. So if you are a curator in DisProt, you will need to log in by clicking this button here. What if you want to explore the resource as a curator to find out if a protein is already there, is already annotated in DisProt, or if it's not there yet? So now you can see that I am logged in because close to this button you will find my name. So to navigate and explore the resource, you can look at the browse page of DisProt and use this drop-down menu with search fields. There are several of them that can help you in performing your research. If you don't find a protein in DisProt and you want to add a new protein entry to the resource, you will see that there is this button here, Add New Entry. You can click it and you will be able to add a new protein entry to DisProt. To do so, you will be required to provide a UniProt accession number. How the curation work is credited in DisProt. In DisProt, we use APICURON. Here you can see the link to the resource APICURON and also the screenshot of a profile of a DisProt expert curator, Maria Victoria. What is the purpose of using APICURON for crediting the curation work in DisProt? We want to value the curation effort and work that each curator does. And we want to recognize this effort based on the accuracy and data quality provided with the annotations of each curator. So the terms and the scores that are available in APICURON reflect the importance of the data that you add through curation. The curator's effort is calculated with different scores based on the type of data that you're going to add to APICURON, the type of information. Moreover, we are going to add soon to APICURON the participation to training sessions for curators and the participation in thematic datasets. So this is an example of the scores related to terms that you can find in APICURON for curation activities in DisProt. For example, the addition of a new publication will give a curator 50 points. The addition of a disorder-related gene ontology function, the addition of a disorder-related IP ontology function will give a curator 30 points. Same way, the addition of a technique used to assess disorder from the evidence and conclusion ontology will provide 20 points. Also, other informations, for example, MIADI, so additional information that can enrich an evidence, will provide the curators with additional points. We have a technique that is frequently used to assess disorder, but not really informative, and very, very common, which is X-ray crystallography missing residues from PDB, that has a score of 10 points. So this is the technique, the experimental technique with the lowest score in APICURON. Which are the essential information for curation in DisProt? So a curator has to start from a peer-reviewed article that contains scientific experiments. That might be scientific experiments that assess the presence of intrinsic disorder in a protein, or experiments that assess a function of an intrinsically disordered protein region. And then we need to know which is the UniProt accession number and the organism of the protein that we are studying, that we want to annotate in DisProt. We need to have the experimental methods details, so which technique or which techniques are used in the publication to assess the presence of intrinsic disorder or one of its features. We need to know the protein region boundaries, so the start and end position of the disordered region. And we will be required to annotate all data of intrinsic disorder using IDP ontology, gene ontology for what concerns functions. Then, for each new annotation that you will add to DisProt, you will be required to add statements from the publications that support your findings, support what you are annotating. Let's say statements from the methods that describe a region as disordered, might be from the results or discussion as well, or statements that describe a function of an intrinsically disordered region. In the case that a statement from the publication is not informative, not clear enough for a user to understand why you annotated that region, you are allowed to add a curator statements. So it means a statement that you can draft in order to make more clear what is happening in your annotation. What is the disordered region? What is its function? Why the data curation in DisProt and in every other manually curated resource has to be rigorous, because this allows for accurate and comprehensive representation of data. And most importantly, it facilitates data accessibility and reusability, because it's an annotation that uses standardized and reliable information. Here you can see an overview of the curation process in DisProt. A curator will start from a candidate protein or candidate publication, will identify the UniProt accession number and create an entry. Then she will be able to add the PubMed identifier of the peer-reviewed scientific article that the curator wants to annotate. The curator then can add the region boundaries, starting then the amino acid positions, and the technique used in that publication to assess a disorder state, annotation, a disorder transition, a disorder function. The technique will be annotated using the ECO terms, evidence and conclusion ontology terms. In the case of disorder state annotations and disorder state transitions, we will use IDPO terms for annotations. If we want to annotate a disorder function, we can either use the IDPO terms if it's a disorder-specific function, so a function that pertains intrinsically disordered proteins only. Otherwise, we can use gene ontology terms if we want to annotate a molecular function, a biological process, a cellular component pertaining that intrinsically disordered region. As mentioned before, we are going then to add a statement or more, if available, from the publication that support the experimental findings that you are annotated. Finally, you could also add additional information, maybe about the sequence construct, experimental condition, experimental setup with MIADI standards that we are going to discuss in one of the later parts of the training. Finally, on this slide, for those who want to have a general overview of IDPs and also a general overview of experimental techniques on IDPs, you can find a list of well-known publications that can help you gain a more broad knowledge of intrinsically disordered proteins.